Okay, all right. Thank you. I don't have any slides, so we're all just going to talk to each other. But I wanted to thank Hugo, Leah, and Tim for some very exciting talks on 3 different but related aspects of what I think is a very exciting area of trying to really figure out how the genome functions, because I think that's where our greatest need is. So we've been tasked to discuss 3 separate questions. So the first is, how should we be doing all of this in the future, which I think is valid. What resources, technologies, computational capabilities do we need to generate and make use of this functional data? And then what do we need to do to overcome the barriers for performing functional studies at scale? So I think what we'll try to do is stick to each of these for about 20 minutes. Otherwise, we'll get stuck on the first for the entire hour. And I'd like to call from people all around the room, especially on the sides, and the younger crowd in particular. And then when you're going to speak, please identify yourself, your name, and where you're from. So for the first question, I want to figure out how should we be doing these types of studies in the future. And I think the types of questions that we should discuss and try to get some clarity on are, how much of this should be done in cell-based assays versus an actual organism? Is the cell the best way to get at what a phenotype is? How do we deal with single gene versus multigenic phenotypes? What do we do about redundancy? How much of this should be done in human versus model organisms? And how much of the focus should be on understanding human biology versus just general biology of the genome and how life works? And then when we're getting into gene functions, I think we should also discuss how much of these types of experiments should be done in single genetic backgrounds versus diverse genetic backgrounds to look at how gene background really affects phenotype. And then finally, how do we prioritize looking at genes, functional elements, and then, dare I say, the dirty word of isoforms. So we can open it up to discussion if people want to address that. They can ask questions, make comments, ask the speakers. Don't be shy. All right, over there.
So Emma Farley, UC San Diego. So I think this was a perfect example of going from reading to writing the genome. And in terms of where do we do this, I think we can get the most phenotypic data if we can. Cells are great, but I think we need to find ways to do these in model organisms. And I think that means also not keeping to model organisms that we necessarily have used in the past, but model organisms that allow us to do these types of research. That's my comment.
Okay, thank you. Bing.
Bing Ren from UC San Diego also. I think these are all very important questions, but I think one of the difficulties addressing them is the lack of clear definition of function. And we all should probably remind of ourselves that what we are looking at and what the conceptual framework that we expect to accomplish. For regulatory sequences, we have some kind of general understanding of what kind of function they possess. And we all know that it's an incomplete understanding, but put that framework out there will help us set the end goal and then define model systems or approaches to achieve that at the fastest pace. Similarly, for protein or other non-protein coding genes function, we probably already have a framework, whether it's kinase, transcription factors, and structural proteins, and then use that framework to define our end goal and march towards it. And also probably, while we're on that topic, we probably should also leave room or technologies for technologies to uncover unexpected functions. So that's my 2 words.
Yes, I think that's a good question, is what is function? Sure. Go ahead.
Alvaro Montero from African Cancer Center. I think one of the things to keep in mind with model organisms is that we've used yeast for a long time, and when we're trying to model mutations from humans, there is an issue with temperature, in which model organisms that have temperature much lower than 37 degrees allow for the buffering of some of the mutations that would be otherwise pathogenic in humans, and we don't see them in model organisms.
Yep. In the very back. Oh, Mark.
Hey, so one thing I'm just curious is now that some of these assays can be scaled genome-wide, how do you think about the advisability of doing them versus more traditional, say, ChIP-seq or chromatin experiments, in terms of cost and what you get for them and so forth?
Okay. You guys want to comment?
Yeah, I can comment on that a little bit. I mean, I think that's one of the things that we need to start to sort out, right? So certainly the information, for example, from a high-throughput reporter assay or a CRISPR assay is complementary. The correlation between that and, say, ChIP-seq signal is not great, which could be surprising or could be technical. I think that needs to be worked out, and I think, right now I don't see it as a replacement, but really something that can go hand in hand as we start to march forward and understand what are really the key components that we should be looking at in gene regulatory function.
Carol, over there.
Hi, Carol Bolt, Jackson Laboratory. So I really have two things. One is I'm really glad that that Hugo and to some extent Tim mentioned sort of the culture or the community that it's going to take to make advances in these areas in functionalization of the human genome. It really is going to take bridging between basic scientists and clinical scientists. The Genomic Medicine Division of NHGRI actually held a meeting on this bench to bedside and back to bench again, this virtuous cycle, and published a paper on it in Cell a couple of years ago, and it really commented on a number of the key areas where we have barriers that we need to overcome to make these collaborations possible. But I think strategically, that is going to have to be one of the areas addressed to really make advances in this area. And then the second one has to do with model organisms. So I completely agree with the viewpoint that we shouldn't stick to the sort of the 6 main model organisms, but I also think that there are a lot of innovations happening within those 6 main model organisms. So it's not just, for example, knocking out a gene in a single genetic background that gives you true functional insight into human genes and variants. It's using some of the emerging diversity panels that allow you to look at the genetic background, the contextual framework for how a variant or a mutation exists and what the phenotypic effects are. So we need to get much more sophisticated about how we use model organisms and also understand their strengths and weaknesses. But I think there's a lot of room here for using the existing organisms in new contexts.
Yeah, I would agree on that completely, and I don't know if Hugo has any more comments on that community-based issue, or even how, when you find a gene that you want to study, which model organism do you pick to study it in?
Yeah, usually it's based on tools and availability of reagents and obviously conservation. I mean, if the gene is not conserved in flies, we're not going to work on it. But the tools are very important, and you can look at the different tools that are available in the different model organisms. They're really very different, and the timelines are also very different. So it's really, you need to sit down with the worm, the yeast, and other people and really brainwash what the best approach is. That's the fastest and the cheapest. And the basic rule is to go with, when a gene is conserved in yeast, ideally you should start there, because that's easy and fast to do, and then you can work your way up the scale and see if it's conserved or not. But I think this is the simplest approach. But given the funding situation right now, most of our efforts are focused obviously on worms, flies, and mice, and some zebrafish work.
Okay. Carol, did you have a response?
Yeah, just that, one of the things that was mentioned, that Hugo mentioned specifically, was sort of the bioinformatics challenges in pulling together all this information. And specifically for model organisms, there is an initiative underway called the Alliance of Genome Resources that is seeking to come up with common programmatic access to the data so that individuals who are interested in this kind of aggregation don't have to go to each individual database, but can go to sort of a common, unified place to get all of the relevant data, where the phenotypes have been consistently annotated and where there are rational mappings to human phenotypes and diseases. So I think there are activities underway that will facilitate the kind of work that was discussed, and it's not new stuff. It's stuff that's been underway that needs to be continued if we're going to make inroads into this.
Over here.
David Valley Hopkins. As a physician who has loved biology and been interested in model organisms all my career, I thought it's been great over the last day and a half to hear many references back to the value of model organism systems. My impression is, and maybe I have a small n, that this enthusiasm for model organisms is currently not as widely shared among study sections as we've been hearing for the last day and a half. And I mean, these are not my own grants, but I've seen grants of my colleagues, and I've been amazed at some of the negative comments coming back from study sections about, well, it's okay to study this in yeast, but yeast are not humans, and what does it really mean, and stuff like that. Comments that I think are really reflect a rather naivete in regarding how biology works. And so I just wonder if that's just a small, my small window, or is there a general need to educate our colleagues, particularly those that sit on study sections, about the value of model organisms?
Yeah, I would like to briefly chime in here. We've seen this a lot, obviously, and sometimes we get comments back, like I have a grant that's in review now to generate reagents for the fly community, and one reviewer said, Drosophila is not the future, I'm not supporting this. And it's really bizarre because it's clear that, you know, we've driven with the research numerous pathways and discoveries, and people seem to ignore them over time. So, you know, you start working, you find a pathway, and then I go to a meeting, and it's the wingless pathway meeting, and people talk about wingless and cancer, et cetera, and there's not a single word mentioned about Drosophila. So the memory is very short. And it's something that people tend to forget. And part through the UDN now, when we started working with the Undiagnosed Disease Network, the first reaction was, you know, how useful is this going to be? But when you start working with the physicians and you start showing how useful it is, they start changing their mind pretty rapidly. So it's kind of having a direct exposure that makes a difference. And now we're really swamped with submissions from physicians. So I think it's just a matter of, you know, turning the tie around and just showing how useful it is and be practical about it. But if you have not been exposed to it, it's a recurrent theme, and it's a complaint that a lot of fly and C. elegans and yeast people have, obviously. Everybody wants to focus on just one organism, and that's the mouse, because they think that's the best. And then you hear a lot of physicians saying that's a terrible model for my disease. So there's no perfect answer, right? We need to work on all the model organisms and do it systematically.
Yeah, I would like to strongly second all of that. Yeah, go ahead.
Yeah, my impression is that when someone says that's a terrible model for my disease, they don't understand their disease.
That's true. Yeah, and I mean, I think not only are the model organisms outstanding avenues to study things, but I think these community resources, things like Hugo does, making knockouts of every gene, I mean, those are such fabulous resources for the entire community, whether you're a fly biologist or mouse or worm, just to be able to order these things and instantly have them made. I just wholeheartedly believe this is a great area that should be supported. Okay, maybe one more comment on this area, and then we'll go to the next one. Haven't heard from you.
Clement Chow, University of Utah. I think everyone agrees that model organisms will be important for all this work to move forward, but I'd encourage NHGRI to continue to support the model organism databases, because that's where we draw all our knowledge from. And if support is cut from those databases, that sends a message to physicians who feel like this isn't important work. that even NHGRI doesn't feel like it needs to be supported to its full extent. We depend on these databases, and if we want to move forward with model organisms, we really need availability of this data to the whole community.
Yeah, I would agree. And as a member of the advisory council, I say we talk a lot about this at council meetings. So it's very well known within the Institute that these are important resources. Okay, so we can move on to sort of the second question now, which is what resources, new technologies, and computational capabilities do we need to generate and make use of functional data? So I have a few ideas on this. I actually think I would strongly support, I think something that would really advance the field would be some sort of initiative along the lines of the $1,000 Genome Project, but focused on DNA synthesis to maybe make an order of magnitude longer oligos that you could use in these massively parallel studies. I mean, if you can go from 250 bases to 2.5 kb, you're in the realm of being able to synthesize every single ORF or most of the ORFs for organisms, and doing things like that would really change the scale and the types of experiments that could be done. There's also technologies that, maybe you could develop things to monitor or sequence all the proteins, RNAs, and metabolites in single cells at the same time. I know I'm dreaming. The genome editing techniques that Tim was discussing, and epigenome editing. And I would say there's another area of RNA editing, not in the sense of ADAR editing, but manipulating RNA in the way that you want to make isoforms. So we can open this area up to discussion. So Joe?
Just going to state the obvious. All three speakers, what they described was really excellent, depended upon technology development. Every single talk, whether it be the variant complexity, the model organism manipulations that can't easily be done in other systems, or the epigenome editing. So I think it has to be in the portfolio of NHGRI to continue technology development, right? Because this is what's driving biology. It's a continuous cycle. You can't do the next experiment in biology you want to do unless you can write a piece of DNA that's 2 kb. What about recording things in cells? So the life of a cell during its entire development that you could record, just like bacteria record what phages saw. What were the genes that were expressed during the entirety of the history of that cell as recorded, whatever. These things are beginning in the community, but I think that should be heavily supported.
Yeah, I would agree. In the back.
As someone who tries to integrate and analyze a lot of data together, I think it's really helpful when we can measure more than one thing on a sample or a cell at a time. So I would make a push for multimodal technology development instead of one at a time and then try to pull them together at the end. This is hard because a lot of the assays are destructive, so I understand it's a big ask, but I think this would be phenomenal. And some of the sort of imaging-based things where we're seeing lots of things in a microscope at the same time look like a promising kind of complement to genomics, which is a bit more challenging to do multimodal.
Yeah. And can you introduce yourselves as you speak?
I'm so sorry, I should have done that. Katie Pollard, Gladstone Institutes, UCSF, and Chen Zuckerberg Biohub.
Okay, thank you.
Can actually.
And Karen. Oh,
I just wanted to, I think that's one thing we did miss across our talks is there wasn't any discussion of sort of this very high resolution microscopy, which I think is pushing the field in incredible new directions and also should be continued and expanded.
yeah. All right, Karen.
Thanks. Karen Miga from UC Santa Cruz. Essentially, my understanding of genome biology and function is really shaped right now by what we can map and what we can target precisely with CRISPR. And I feel like all of us in this room clearly understand there's a place at the table for function for repeat biology. This has been brought up continuously through this meeting. However, I would love to see in the next 10 years some new technology development to actually bring these into focus. And I think that there's no question that repeat biology is involved in some of these high-C looping. They're involved in some of the gene regulation models that we have. However, the tools just simply aren't there, and they're not being discussed actively. And with the comments of kind of synthesizing DNA, I definitely see the promise there, but I feel the worry for me is that we're narrowing our focus instead of broadening it. And so I'd just like some advice from the panel on where you think the next 5-10 years is for including this kind of ignored portion of the genome.
Anyone?
Sure. Yeah, no, I think these are great points. I forgot to mention in my talk, as we're synthesizing, where does synthesis get stuck? It gets stuck on repetitive regions, and you can't synthesize them, right? So I think it's a massive challenge. I think, there is some interesting advances. So I mentioned, when you're doing assays, for example, on patient DNA, you don't need the genome sequence. You can look directly at that. I mean, I think there's still a, sort of sequencing to observe the repetitive regions is critically important. But I think there are some emerging technologies to assay them. Yeah, I mean, I think there's huge potential in here that is untapped. I mean, triplet, expansion repeats and promoters screwing up gene expression in all sorts of crazy ways. I think a lot of this is kind of my comment on false negatives, right? That we're going to miss a lot with what we have now. And I think we need to be careful on how we interpret that and how we think about that as an opportunity, not as a, functionally deficient region.
Yeah, I would agree with that, and I would just say, I forget who it was since I was watching by webcast yesterday. You all look great, by the way, on TV. Somebody wrote, in the next 10 years, somebody will synthesize the first human chromosome, but how are we going to do that if we don't even have the sequence of a whole human chromosome yet, let alone in multiple people? So I think we need to fill that gap for sure, and it should be, it's part of that hard to address area, but it needs to be addressed because it's essential. And we don't even know how important it is until we have that information. So, oops, over there.
Judy Cho, Mount Sinai, New York. So this is under the computational needs. We kind of think of genes as actors, but again, when you think about, systematic annotation of variation, you want to think about variation that causes change in their capacity to be acted upon by other genes in terms of like kinase substrates, glycosylation substrates, and proteolytic substrates. So again, can that scale? These are the overexpression can give you false positives and so forth, but I think that's an important need.
Okay. Ali.
So the way I think about the human genome compared to, say, Drosophila is that there's a lot of redundancy in the human genome. And so I wonder if you guys are confident that when you do all of these surveys with CRISPR and so on, you're really able to capture how the system is going to deal with the redundancy in there. You might see a 25% change in the activity of an enhancer, which may at the end of the day not lead to any change to the RNA and the protein that's going to be staying around, which will then affect how the system responds. I don't think we're right now able to predict which genes are going to be affected by a change in expression level that's subtle. You can predict 100%, right, a complete knockout, but even haploinsufficiency is a tricky thing. So I expect the problem will be worse with enhancers, and so I'm just wondering what we can do to sort of include the modeling of the feedback into all of these things.
Yeah. In the fly, we have the same problem. I mean, there's quite a few genes for which there's 2 or 3 homologs. I mean, and we have to deal with that. In fact, we're doing this now by doing systematic double RNA knockdowns by creating transgenic lines with 4 guide RNAs, 2 guide RNAs for protein 1 and 2 guide RNAs for protein 2 to hit the 2 different genes, because there's over 1,500 genes for which there is no phenotype, and there are clearly 2 homologs. And so doing a systematic study to find how redundancy is revealed into the genome is something we're really tackling right now. And this is a project with Norbert Perimo. The second issue is that when you're looking at the human genes and you put them in fruit flies, we have quite a few cases where, for example, for EBF3, there is an EBF1, an EBF2, an EBF3, and an EBF4 in human. There's only 1 transcription factor in flies. All 4 human genes rescue the fly gene. So that's a simple case, because that single mutant in Drosophila has allowed us to discover 4 new human diseases and variants in all of these 4 different genes. On the other hand, sometimes, redundancy is bizarre for mitofusin, for example, for which there's 2 homologs in human. You need to express both in Drosophila to rescue the homolog in Drosophila, showing that clearly in evolution these 2 proteins have gotten different functions because you need the 2 homologs in the fly to rescue a single mutant. So there's a lot of variation there, and there's a lot of different issues, and they're not easy to address. It's systematically doing the work that will answer these questions, and there is no simple answer.
Okay. In the back.
Sean Burgess, Genome Institute. I just want to pile on top of this that there does seem to be, although probably not in this room quite as much, this generalized belief that all the gene functions have been annotated in the genome and we actually understand what they do. That's probably true for maybe 20 to 30% of the genes in the genome. And because there's been some big initiatives, it's sort of like, well, it's a settled problem. I think we need to readdress that belief and actually reassert our desire to actually understand what all the genes in the genome, just the genes, forget about regulation, forget about everything else, what the actual genes themselves are doing. And that includes not, our attitude has to change from, genetics, it used to be A led to B led to C, and now we're realizing it's networks and complications. We also have to realize probably many, if not most, genes have more than one defined function. P53, what's it doing in the mitochondria, right? So, so maybe even a systematic subcellular localization of all the proteins in the genome would be an essential part of understanding those functions.
Yeah, I couldn't agree more. When you look at all the databases from all the model organisms, you see that there's so many genes for which there is essentially no information. Just to give you an idea, I got a list of 24 genes from a Chinese hospital where they've tried to identify new human disease with one phenotype, early onset epilepsy and encephalopathy. And they gave me these 24 genes. All of them are unstudied. Every single one of them. So, there's a lot of work that remains to be.
Okay, great. There's also isoforms, all the isoforms. Okay, Tuli.
So now that the current phase of ENCODE is coming to an end, we've obviously gotten quite a bit of data about transcription and splicing factor binding. But these are the key sequence-specific regulators. And as we want to learn to predict regulatory effects of genetic variants, understanding just the function and binding motives of these key regulators that are really sort of like fundamentally every regulatory variant has to somehow affect some sequence-specific regulator sometime during the cell's epigenetic lineage. And the catalog of these that we have and actual ChIP-seq data, actual assays for TF and SF binding is very, very incomplete. And I think that this is something that just needs continued investment going forward.
Yeah, I'm biased, but I completely agree with that. I think every DNA and RNA binding protein needs to see its day in the sun. All right, there it is.
Hey, Erez Aiden, Baylor College of Medicine. So I wanted to just kind of remind folks of something that people have alluded to, but hasn't really been directly discussed, which is that for a long time, these kinds of questions of trying to explain the function of all sorts of variants was thought that you'd be able to do this using the formation of loops in 3D. And I think one of the things that's happened in the last few years is we've become very good at actually mapping the formation of 3D chromatin loops. This is something that we should continue to exploit. I do say that with the sort of cautionary addition that I think this is not a silver bullet. We have to do this incredibly carefully and make sure that all of the results are reproducible and valid, et cetera. But I think that this is doable, and I think at least some piece of this problem of how variants come to affect particular genes will be explained using these methods. So we should complete that thought. I also wanted to echo Katie Pollard's point, which is that I think microscopy is going to become an incredibly important platform for genomics in the coming 10 years, and we should keep that in mind.
Yes, great points. Howard.
Howard Chang, Stanford University. I want to thank the panel members and also add to the excitement of these new technologies for really sort of speeding up the scale of functional interrogation. Just want to further add that there are now assays that actually created by many in this audience to actually both edit or modify genome, but then read out the consequence, not just with alive or dead or reporter gene output, but genome-wide measurements of RNA like perturb-seq or chromatin accessibility. And so they give you a lot more depth in terms of the consequences of these genes or elements. So this is an area of rapid technology development, and hopefully should be continued supported by NHGRI, because we can get a lot of information and a lot of exciting advances right now.
Okay, great, thanks. So before we move off this topic, can anybody comment on any techniques we haven't discussed that we need to address that NHGRI should support in the future? Sharon? Okay. Not working? Everybody have their mics off? There we go. Okay, we can go over this. Okay. Anyone? All right. Aviv.
Thanks. So actually two things. The first, which I think generally NHGRI has seen out of scope, but is actually limiting for many of the assays, is delivery. If you want to perturb systems, you have to deliver into them. And I think it's telling that most of what we see comes out of cell lines, because these are easy to deliver to, and doesn't come from primary cells, organoids, and whole organisms, because these are difficult to deliver to. And that's going to be something that we'll have to grapple with. The second item is the combinatorial space. Every time somebody mentions interactions, I think it's important to realize that as soon as you want to think about this problem systematically, in the naive, exhaustive sense, this is not going to be solvable because there aren't enough cells, period, in the world. So that means that we have to come up with interesting and clever solutions. Historically, those meant looking at more limited, targeted areas of the space that data pointed us to. For example, look at the paralogs because you know they might be redundant. And I do think that it's a worthy 10-year goal to try and look for other means by which we can look at more.
Okay, excellent. We have discovered this whole section. Okay, yeah. Use Eric's mic.
I was actually going to say a different kind of combinatorial. I think, and Hugo maybe exemplified it the best, but I think that the goal here is not to have a functional assay that is going to perfectly predict disease. In fact, that's not how genetic labs analyze variants. We combine all kinds of information. So I think the optimal way of combining existing human data with functional data to come up with a conclusion is something, certainly something Klingen's thinking about, but something that requires work. Because Leah showed kind of a live dead assay, but many of the readouts are going to give intermediate values where you're going to have to take that data plus human data to come to a conclusion. And the best way is to combine that information is something that I think needs work.
Okay, excellent. Good point. It looks to me like anybody on that side of the room might have to come. Oh, are they working now? Okay. It was a Christmas light special over there. Jay, did you have a comment?
Yeah, this is maybe echoing some of the other comments, and also maybe repeating myself from yesterday. But I think the phenotyping is key here, right? In almost everything. Obviously, getting these assays to work and to scale is one piece of it, but the phenotyping is really important. I think that a focus on molecular phenotypes in particular, I think, is more likely to yield useful functional insights, and as well as to get at some of the things that like Ali raised about redundancy, where you can isolate these things and maybe have a quicker route to I think part of the issue with things like redundancy is that we don't really have a good understanding of it, right? And this is also an opportunity, I think, to attack some of those things if we keep the focus at that level.
Okay, great points. And are there any new resources that need to be generated that NHGRI could take the forefront on?
Nadav Aitu of UCSF. I think it's great everybody's generating a lot of various
saturation mutagenesis data sets, functional data sets, including our lab and others, but I think we really have to have resources that combine all of them together where people can view all of these results together in one place or a few places, but that combine all of these so it'll be really easy for the end user to really see the effects of all these variants and combination of these variants.
Yeah, I think I want to chime in here. This is indeed very important, and this was already alluded to by Carl Bell, where for the AGR, we need to look at all the model organisms. We need to look at all the information that's available for all the variants and integrate that knowledge. And there is a wealth of information there that's really untapped, and it's a very important issue. And the second issue is with respect to the presentation, the integration of the data and the presentation, making simple presentations. We're trying to do that with Marvel, to integrate all of this information and present it in a cogent, simple way, so you don't need to go to all of these individual programs to figure out really all this information. And I think there should be an emphasis on doing that, on developing programs that integrate all the information in a quick and easy way. And you don't need to be a bioinformatician to understand each of these programs. I think vulgarization, in a way, is what I'm aiming at.
Yeah, and I would argue you can't be a bioinformatician to understand it, because most of the people, especially outside of this room, are not. So I think we have to make these things very easy for people to access and to collate things together. Is there somebody over there? Yes.
Yep. Ting Wang from Washington University in St. Louis. I want to make two quick comments. One is about transposable elements. It's just like this half of the room, you know, the 50% of genome do not receive enough attention. All these nice functional assays, you know, genome-wide CRISPR or, you know, parallel reporter assays, transposable elements are deprioritized, right? So, you know, for good reasons, they're, you know, repetitive, but I think in contrast to, you know, what sort of the common practice, I think they actually deserve more attention, more specialized attention in terms of developing technologies as well as resources, both experimentally and computationally, to directly tackle this part of the genome. So that's a comment I like to make. The second one is to really echo what Bing mentioned earlier, and then to a large degree also by Ali. That is the definition of function, because without that, we won't be able to explain or understand what is non-function, what is a negative result. I mean, you take out the enhancer, you don't see a measurable transcription output, and then for many people, including my own lab, you know, that often to be the end of it. Reviewers just, you know, shut you down right there if you don't see a transcriptional output. I feel that we need to have a more systematic way of discussing and understanding what does that result mean and how to really comprehensively measure function. But before doing that, we need to define function and, in that context, non-function. Thank you.
Okay, excellent points. So in the sake of time, we should probably move on to the third question we've been asked to address, which is what is needed to overcome the barriers to performing these types of functional studies at scale. And I think if Aviv's point of there are not enough cells in the universe is potentially an issue. So how do we deal with this? So, Ross.
So I want to, as we get into that, I'd like for us to be clearer about how high are these barriers that we're confronting, and in particular, let's just think about assays for regulatory elements. Tim Reddy did just a great job of surveying the many different procedures, and you'll remember that some of those, and the way StarSeq was developed, was to cover the whole genome, right? But there are many examples that you know of, and Tim went through several of these, where there's a focus on transcription factor bound places, or, you know, we're talking about developing these catalogs of candidate regulatory elements. And should we stick with genome-wide for the regulatory elements because we don't know everything? Or, and my perspective is, we do know something, it's not insignificant, and that has led us to develop these biochemical assays that we're all familiar with, and it's highly useful, and to me it was kind of a no-brainer that that you do that focus on regulatory elements, but some candidates, the things that you develop by doing these assays and by doing integrative modeling of them to try to predict function and focus on those. But I think it might be worth stepping back and asking, are we going to try to be comprehensive? The assays that Leah was talking about, let's make all the substitutions, and saturation mutagenesis has been really powerful. But frankly, I don't see how you can do that genome-wide for regulatory elements, unless you say, hey, there's these 10 loci that we think we could focus on and do it really deeply. So that's my comments. But the question is, and to the panel, is can we make a strategic decision now? Have we learned stuff from the ENCODE functional characterizations, which I think have both activities going on, that might guide us?
Yeah, so, right. So, Ross, I think these points are excellent in that, there's going to be a really powerful balance between being comprehensive in some areas and being very focused in others. I think there's some interesting work going on now in trying to find a middle ground between those. I think this work is starting to be developed in many different groups, thinking about how do you sample this space in a way that you can be representative if you cannot be comprehensive, right? So I think there's some opportunities there that could be further developed. It's kind of hard to think about. I mean, in some areas, for example, you can treat cells in infinite numbers of different ways. We don't have a good sense, I don't think, of what the range of environmental responses could be, right? So I think there could also be some interesting scoping studies in trying to get a sense of what the boundaries are, so we know if we're going to sample across this space, what that actually looks like. And I don't know if that's feasible or not right now, because I don't know how big that space is, right? Certainly the genome squared is gigantic, and I think that's where we run into a lot of the issues that Aviv raises, right? So, when you're talking about combinations of variants, I think, there's some natural ways to focus in haplotypes and things like that too that can help guide us, basically leveraging human populations, evolution, et cetera, to give us a sense of where the important, we don't want to assay every combination of variants if it never is going to exist, right? So I think there's some ways to balance in between these that are forthcoming and need to be focused on.
Any other? Barbara.
Oh, sorry. Barbara Wold, Caltech. So I think the topic of combinatorics has come up several times, and particularly in the regulatory element domain, the place for go relatively deep to scope out how much combinatorics there are, not only within elements saturating there, but among joint elements. It isn't so much explaining redundancy. It's about assaying how a suite of regulatory elements impinges on a gene or a set of neighboring genes. And so we will have to go deep in some domains simply to scope out what we have to do. And if we stick with the individual elements one by one, we will be informed but sorry and only be getting part of the picture.
Yep, good point. Trey.
Yeah, so Trey Eidecker, UCSD. Yeah, so this issue of combinatorics is really interesting, and as it's been pointed out, you can saturate maybe all single perturbations to a system, but once you're pairwise, much less larger combinations, there's not enough atoms in the universe, much less cells to survey them all. And so I think one weapon in this is combination or integration of data types, especially integration of functional data like we've been talking about with biochemical or biophysical interactions. So I was struck in Hugo's talk how the key hook was this biochemical interaction with ankle 2, I believe it was. Maybe I got some of those details wrong. But the idea is that the sort of physical interaction space of a protein can greatly inform what combinations you might then want to test in candidate gene approaches.
Okay, excellent. Aviv, again.
Just because I triggered this. So I would challenge us I think there are two aspects to the solution to the not enough cells or atoms or whatever in the world. The first is the one that I think people have been triggering already, which is use the data that you have to know where to search. That's a way of limiting the search space. The other is to come up with more efficient ways of doing experiments compared to what we do today. And I think some of them might feel initially like antithetical to the idea of you always measure everything, every gene, every molecule, everything you can measure because that's what genomics does. And it would be a big transition for us in the next decade if we want to go after that scale, after that combinatorial scales. And sometimes we will be doing it, I think, in ways that are not yet fully imaginable by us. So that's why it's an important stretch goal. If we already knew how to do an experiment like this, then it would be a great target for the next 10 years. It would have been a great target 10 years ago.
Yep. Yes, Andy.
I'd just like to point out that in a way, these experiments that Aviv is asking for are being done all the time. They're being done all the time by nature. The human population is, in fact, a saturation mutagenesis experiment about 50 times over every generation. And that evolution is very, very good at detecting some functional differences. So I'm thinking particularly about Daniel MacArthur's NOMAD project, and by lining up the sequences of sufficiently many humans, you can identify regions of proteins that are absolutely intolerant to variation. And so I'm wondering if this sort of application of evolutionary reasoning could be scaled up in various creative ways. For instance, doing something like NOMAD in mouse, fly, worm, to identify, what regions of the genome are intolerant to variation, to make much better use of evolution. So we all have a very kind of naive idea that, okay, if it's functional, it's conserved, so we just look for matches. So if the gene is present in some other organism, it's there for a reason, it must be conserved. But evolution can give information that's much more subtle than that. For instance, we can identify distinctions between functional relevance of paralogs within a genome by looking at many sequences within a species instead of just conservation between species. We can, through distortions of the site frequency spectrum, see relative importance of regulatory regions, protein coding regions, and so forth. So I think there's really a lot of opportunity for a kind of intersection of scaling up the resource in terms of quantifying variation, the informatics tools that line up with human disease, and the use of these tools for selecting what's the ideal model organism for a particular question.
Yeah, I think that's a really great point. And again, I think for resource-wise, I would say sequencing the genome's high quality of as many organisms as possible would help get into that as well. Aviv is dying to respond.
I'll just answer Andy and then I'll shut up. I think this highlights the point that we've been thinking of screens in cells and models and one cell type, but we should be thinking about these high content readouts in an experiment already done by nature in 7 billion humans. And that is a resource question because you need access to things that you don't just genotype, but you also phenotype at a molecular scale that allows you to do genetics. So we need to think constantly in both ways, both in the lab, what we can do with models, and what we can do with humans.
Okay, great. Eric.
So let me bring us back to the things where there are enough atoms in the universe, because the combinatorial really is amazingly interesting and important and is still at the stage where we're piloting and feeling our way around. I want to bring us back to where we can truly build comprehensive resources. Because I think one of the great things NHGRI has done over the last 30 years is identify those things where we can truly be comprehensive. Get every nucleotide, get every common variant, get a comprehensive picture of the haplotype structure, and I could go on like this, where every means, almost every. There's always some little stuff around the edges. So I'm curious, where can we be truly comprehensive? Now, comprehensive refers to both everything in the genome, if you're talking about non-coding elements, all non-coding elements, or all protein coding regions, but also with regard to the functional assays. A functional assay for your favorite property is very good if you're studying BRCA or this or that, but of course, things like total gene expression Might be the case that if you do the protein mutagenesis experiments, you put in every possible allele, whether something looks like the null allele or the wild type allele at the level of whole gene expression, might be a fairly sensitive assay. And in any case, you might pick intermediates that group in some ways. Look at everybody, not everybody, look at hundreds of people and do whole human cell atlases on hundreds of people, so that you get an EQTL catalog for every cell across the whole genome down to a certain frequency. I think one of the things we have to do, and I'd be interested in the panel's thoughts, is identify what in the next 5 years do we think can truly be reduced to comprehensive catalogs. Because there's a huge difference when you're comprehensive. You know that there's no variant lurking in the bushes you haven't seen. And then there are other things that are still at this pilot stage or bespoke to a particular disease. But if we came away with 4 or 5 targets for the comprehensive foundation, even if they're very ambitious but just not insane in the sense of requiring more atoms than there are in the universe, I think that would be a great thing for this next 5 years.
Yeah, I think that's a great point. And I think one of those things that we could be comprehensive is to finally finish the genome. So we have telomere to telomere sequences.
But I want to note, that's exactly the way in which I'm not using comprehensive. So that by comprehensive, there's to go from when you got 5 or 10% to when you got 90-odd%. Because when you got 90-odd%, you usually have it there. Then there's absolute complete finishing. That's a great thing, but it's usually extremely expensive, and it usually doesn't radically change what you're going to find. I'm in favor of it, but that's not what I'm talking about. I'm really talking about comprehensive, when the genome got sequenced, when we got to the point where we had 99% of the euchromatin, for all practical purposes. So I want to be clear that when I say comprehensive, I mean, for practical purposes, we have the vast majority of it. For most of these methods right now, we're nowhere close. We're at the 5% level. Get us to the 90% level, 95% level, and then we can have separate things for completion, but those tend to be different and expensive and have different properties. So I'm in favor of both, but for these kind of non-coding and coding assays, to move from the examples we've got to essentially knowing that you could look it up and almost always it will be there is a huge transition that the next 5 years could get us.
Yep.
I don't even.
Back there.
Yep. Hi, John Mudgett. So one key takeaway from the IDG effort of the NIH, the Illuminating the Druggable Genome effort, was they approached how to tackle whole families of genes and scale the functional outputs, and to take a concept in the scalability of functional assays. Now they approach it as families, as kinases, as GPCRs, et cetera. But if there are lessons learned in taking functional assays and scaling them up by family, perhaps understanding how they approach that and leverage that would give some information back on other families which are also interesting to this room.
Okay. And Mark.
Hi, I just want to second Andy Clark's comment about evolution again, and I just did want to point out that, one of the nice things about sequencing larger and larger numbers of organisms in people is that there's a very straightforward path to calculating additional things. For instance, if you sequence enough things, you can look at pairwise interactions between things and get connections between things, and it's very straightforward. It just requires more data, and I think that's a nice thing.
Okay. Any other points? Yes.
Andrea Califano, Colombia. So I wanted to speak a little bit to what Eric just said, and I agree wholeheartedly with it. However, I think there's also a risk. So if you're trying to compute the time it takes for a train to go from New York to Boston, a comprehensive approach by physicists would say you have to use quantum chromodynamics, and that would be a very inefficient way to actually compute that time, and normal Newtonian mechanics will do a much better job of it. So I think that there is a tremendous value in creating comprehensive repertoire of information, of data sets, regulatory elements in the genome, et cetera. But it would be actually, and this is something that I think has been missing on HGRI, it would be actually better to do that in the context of a specific phenotypes or a set of chartered phenotypes of interest, because what we've learned from other areas of investigation, cancer and neurodegenerative diseases, et cetera, is that if actually focusing your attention on the particular phenotype will actually highlight certain mechanisms that are critically relevant to integrate the effect, for instance, genetic alteration, leading to essentially the same phenotype, regardless of what specific one of the quadrillions of patterns of mutational alteration may actually be at play. And I think focusing on those mechanisms, very much as is happening in normal homeostasis, for instance, is going to be, and it's different from phenotype to phenotype, is going to be a really important way to figure out how to actually build these repertoires of comprehensive information.
Okay. Yes, Howard.
Howard Chang from Stanford. I want to riff on Eric's comment. I think it's a really interesting idea, and I was thinking about maybe one of the ways we can gain a lot more insights is to combine what we're talking about in this session with the sessions from yesterday about variant discovery. What if we actually did all these molecular assays, the deep phenotyping that we're talking about, from samples from which we're actually doing our variant discovery? That is, these are individuals that we have actually clinical phenotyping. We're doing a lot of this genotyping. Then we have the cells from them to actually do these actually functional protein variant analysis or, let's say, non-coding element studies. Now, if we're thinking about that, agnostic as to what disease we're talking about, this actually requires some resources and differences in support in organization that NHGRI can help with. Because my understanding is that these association studies, oftentimes all you have is the DNA, but not even nuclei that we need for some of our functional studies, or even cells that you can expand and grow. Okay, and so that would be, now some of these assays we heard about, they're very, like this haploid assay is great, but it's a very sort of, let's say, sort of specialized and weird cell line. So we have to be able to expand cells from all these clinical biopsies. So this is typically not in the remit of NHGRI, but would be necessary if we want to think about these kind of large-scale projects.
Okay, excellent, thank you. All right, John, and we only have 2 minutes left for this session.
John Stamford-Anopoulos, University of Washington, Altius. I just want to make a comment that, I mean, one word that has come up, or a couple of things that have come up repeatedly are comprehensiveness and also scale. And I think one of the challenges is that one of the lessons we've learned in the last 20, 25 years over and over again is that if you have assays that perform okay but have some problems that you maybe swept under the rug while you publish the paper, when you take that and scale it, those problems overwhelm the entire system. And I think, you know, having a really clear understanding of the performance characteristics of various assays. I mean, sequencing, we understand ACGT, we're pretty confident that these calls are correct. When we look at large-scale CRISPR screens and we neglect off targets and we don't actually measure genotypes, we're just sequencing guide RNAs. There's all kinds of things that may be in the system that we're not really understanding carefully. So I think understanding, having a clear focus on hardening the assays is going to be essential to addressing both comprehensiveness and scale. And that seems to have been sort of overlooked.
Yes, absolutely. Okay, any final comment? Yes, back there.
I just wanted to make a specific proposal that we focus on transcription factor DNA interactions. I think that would be an area where we could be comprehensive. Sorry, Adam Siepel from Cold Spring Harbor. Area where we could be comprehensive, and it would focus many different efforts from EQTL to understanding epigenomic modifications. I think it's very much in NHGRI's wheelhouse as well.
I completely agree with that, but I would double it and say all the RNA binding proteins too. So, all right, well, thank you. This has been a great discussion. I'd like to thank the 3 speakers and all of you, and I believe we have a break now. Actually, I have a okay, Elise needs to talk.
Okay, thank you all very much for a great set of presentations and discussion. So we thought logistically it makes more sense to give a bit of the additional guidance for the breakout sessions now, and then you can break and then make your move to the specific breakout sessions you've been assigned to. So I just want to remind you of the three sessions for today. The breakout session 4 will be identification and characterization of all genes and regulatory elements. Breakout 5 is determining the functional consequences of variants acting individually and combination. And then 6 is accurate prediction of the regulatory consequences of variants and modeling gene regulation. I just wanted to make a couple of points about these sessions. There was a lot of discussion with the co-chairs and the moderator for the session about really how are these distinct. We tried to provide some guidance about those in the breakout session guidance document where we gave examples of what topics could be covered under each of these. These are meant to be examples. They are not meant to be narrowing down, but hopefully are clarifying. These are also different ways of looking at genome variant function from different perspectives. We would not be surprised if some ideas came out, some of the same ideas came out of the various groups, and that's fine. But we have a plea for this set of breakout sessions to be come up with perhaps more concrete, specific ideas for projects at the end that we think would be very helpful in forming the next set of discussions. So perhaps you may want to spend the last few minutes of the session in reviewing what's been discussed and putting together your specific thoughts and see if there's agreement on that. We have an additional
tiny little thing, and we're done with the guidance.
I think we're done with the guidance, except I just want to say that please be in your seats and ready to go promptly at no later than 10:30. And 1 hour for the breakout sessions is really short. And actually, I'm going to modify this since it's only 10 o'clock. Why don't we be ready to go at 10:15 and give us a little bit more time in the sessions. And Chris, you have an announcement.
Just tiny logistical thing. Lorgetta Schools has been helping out with doing getting presentations where they need to go. Her email is down right now, so if any of the breakout session chairs have any presentations that need to go somewhere and you need help, send them to me, Chris Wetterstrand. Okay?
Great, thanks. So we'll take a break now, and we'll start the breakout sessions at 10:20. I was gonna say I was shocked. I didn't want to embarrass you.
I know.